بسم الله الرحمن الرحيم. محاضرة اليوم راح نتكلم بيها على ال development of the nervous system to understand the development of the central nervous system from embryological point of view. We will discuss in this lecture the outlines of the embryological development of the nervous system and some associated problems. We will define the neurulation, the process of neurulation and the neural tube formation, then the origin and fate of the notochord, development of the neural fold, neural tube fusion, and consequences of the abnormal defects of this development. Then we will discuss the development of the spinal cord and development of the brain, then the development of the ventricles, alar and basal plates, and the neural crest formation. The nervous system development is the most complex system in the body. It is the first system to be developed and the last one to be completed. Because the length of time taken to develop the central nervous system, the nervous system is most susceptible system in the body for insulting during the prenatal life. A good understanding of how the nervous system develops allow us to deep understand the system in the adults. The first step in development of the nervous system is by gastrulation that occur in the second week of gestation. After the implantation, the embryo will develop and form two layers. That is called the epiblast, the superior layer, and hypoblast, the inferior layer. Then, in the third week of the gestation, the process of gastrulation occurred that changed the embryo into three layers. The upper layer called ectoderm, the inner layer called endoderm, and a third layer formed by the process of gastrulation that is called the mesoderm. In the third week of the gestation, the notochord will form. The notochord is a solid cord of cells formed by pre-notochordal cells that migrate through the primitive pit. Definitive notochords serve as the basis for the midline structures, axial skeleton, and nervous system development. The development of the notochord will induct the neural tube formation. Then the notochord will develop finally until reaching to the full maturation, and it will form the disc between the vertebrae. The primitive pit will form in the caudal part of the three-layer embryo, and from this pit, the cells will migrate to the cephalic portion to form the notochord. And this picture shows the sections of the embryo to see the notochord. And above it is the ectodermal layer. Below it is the mesodermal layer. So it originated from the ectoderm. The notochord will induce the formation of a neural plate by elevation of the lateral edges of the neural plate, then depress the mid-region of the neural groove. The neural folds gradually approach each other in the midline and diffuse, producing the neural tube by this process. This is the neural groove in the midline. The edges will elevated to form the neural folds. This fold will completely attach to each other to form the neural tube. Neural plate formed about the day of 18th day of gestation. Then the neural groove will form at the next day. Neural folds begin to elevate and continue elevation until it reach in the midline. The neural folds fuse to form the neural tube at about the day 21 to 23. By the day of the 28 to 32, the neural tube is completely closed. The whole process takes place in 10 days. Fusion of the neural folds. Fusion begins in the future cervical region, proceeds in both cranial and caudal direction. Defect in closure of neuropores underlies serious birth defects of the nervous system. The openings in the cephalic side will call the cephalic neuropore, and the opening in the caudal part called caudal neuropore. The cephalic neuropore will close earlier than the caudal neuropore. As a summary, neurulation begins in the third week. Notochord will drive the induction of ectoderm that lead to formation of a neural plate. Forming the neural plate will drive the neural tube formation. Folding continue until the closed neural tube is formed with anterior and posterior opening that are called the neuropores. The anterior neuropore close on the day of the 25 and the posterior neuropore closes after 2 to 3 days on the day of 28 of gestation. A neural tube defect results from failure of neural tube formation. If the neural tube will form abnormally, the failure of neural tube formation will occur. That is called failure can occur at the caudal portion, causing spina bifida, or occur on the cranial portion that's called anencephaly. Spina bifida, that's mean opening of the caudal part of the neural tube, can occur anywhere along the length of the caudal part of the neural tube, most commonly in the lumbosacral region. A neurological deficit occur, though not associated with mental retardation, so it is unaffected on the brain development, but it's usually associated with hydrocephalus, that is, increased amount of cerebrospinal fluid inside the brain ventricles, due to many causes like obstruction of the CSF circulation or excessive amount of production of the CSF circulation. This picture shows the 3 types of spina bifida. The first picture shows the spina bifida occulta, that is associated with superficial defects only without affecting the spinal cord itself. The defect in the vertebra only. The second type is spina bifida with meningocele that affect the meninges that cover the spinal cord, causing a cyst formation called meningocele. The third type, which is the dangerous type, is the spina bifida with meningomyelocele that affect the meninges and the spinal cord itself, causing cyst formation called meningomyelocele. Other neural tube defects like anencephaly, which is the failure of the neural tube formation on the cranial part, absence of cranial structures including the brain that is incompatible with life. Another example is rachis, failure of a neural fold elevation will lead to splayed neural tube. These pictures about spina bifida occulta, or spina bifida with meningocele, and spina bifida with meningomyelocele. And this picture shows the anencephaly, which is a very dangerous case. It is incompatible with life. Anatomical division of the adult central nervous system. The brain divided into 3 parts. The forebrain, that is formed by cerebral hemisphere and diencephalon. And the second part is the midbrain. And the third part is the hindbrain, which is formed by the pons, medulla oblongata, and cerebellum behind them. And lastly is the spinal cord. How to make a nervous system? The development of nervous system begins from a simple hollow in the neuroectodermal tube. The cells on the edges of this tube will migrate to form a neural crest. Then lumen will form ventricular system. 3 dilatation on the cranial part will form 5 dilatations that form the anatomical divisions of the brain. And caudally, the tail will form the spinal cord. Development of the brain and the spinal cord then suffer from patterning and organization. The bending of the caudal part of the neural tube will form the flexures. So we will discuss these processes: ventricular system, anatomical divisions of the brain, spinal cord, patterning and organization, neural crest, and flexures. The spinal cord development. Most of the length of the neural tube give rise to the spinal cord. At the third month of gestation, the spinal cord is the same length as the vertebral column. Thereafter, the vertebral column grows faster. The growing of vertebra grow faster than the spinal cord development. So the spinal cord will become shorter than the vertebral canal. But the spinal nerves at the caudal part of the spinal cord must elongate to reach their corresponding intervertebral level, form the cauda equina, the lowest point of the spinal cord forming conical shape, and that is called the cauda equina. At the third month of the embryonic life, the length of the spinal cord is the same length of the vertebral canal. So the spinal nerve will emerge between the corresponding intervertebral foramina. But at the fifth month, the length of the vertebral canal will exceed vastly the length of the spinal cord. So the spinal nerve will descend more distance to reach its corresponding level. At the day of birth, a newborn, the length of the spinal cord reaching about the level of the third lumbar vertebra and form conical shape end of the spinal cord that is called cauda equina. Development of the brain during neural fold formation, the three primary brain regions can be distinguished. At the cephalic part of the neural tube, three dilatation will formed. The first dilatation, called the prosencephalon, that is form the forebrain. And the second one, called mesencephalon, that will form the midbrain. And the third one, called rhombencephalon, that will form the hindbrain. These dilatations called primary brain vesicles. Primary brain vesicles like this picture. The first vesicle, prosencephalon, will form the forebrain. The second vesicle, called mesencephalon, that will form the midbrain. And the third vesicle, called hindbrain, that is the rhombencephalon. In the fourth week of gestation, these vesicles show this picture. This picture about the 27th day of embryonic life. Then, at the fifth week of development, these vesicles will split to form 5 vesicles. The first vesicle, which is the prosencephalon, will divide into telencephalon and diencephalon. The telencephalon and diencephalon. The second vesicle, which is the mesencephalon, still single vesicle. The third vesicle, which is the rhombencephalon, will divide to form the metencephalon and myelencephalon. So the brain formed of 5 vesicles: telencephalon, diencephalon, mesencephalon, metencephalon, and lastly the myelencephalon. These pictures show the prosencephalon, mesencephalon, and rhombencephalon that are called the primary brain vesicles. But splitting of these vesicles for forming the telencephalon, diencephalon, forming the forebrain, midbrain still by the mesencephalon, and the third vesicle, which is the rhombencephalon, divide to form the metencephalon and myelencephalon. The prosencephalon will form the forebrain, telencephalon forming the cerebral hemispheres, and diencephalon forming the thalamus and surrounding structures. The mesencephalon still as single vesicle, forming the midbrain. The rhombencephalon divide to form metencephalon and myelencephalon. The metencephalon will form the pons and cerebellum. The myelencephalon will form the medulla oblongata. So, the anatomical division of this adult central nervous system formed of the 2 cerebral hemispheres that are derived from the telencephalon, and thalamus and hypothalamus surrounding structures that are derived from the diencephalon, the midbrain derived from mesencephalon, and the hindbrain, which is formed by the pons and cerebellum from the metencephalon, and medulla oblongata from the myelencephalon. So, these steps will completed finally 5 anatomical divisions of the brain, and the spinal cord is formed completely. The second step is the flexures. Growth and development at the cranial neural tube exceed the available space inside the cranial cavity. So the neural tube will fold up, leading to two flexures at the cervical region and at the cephalic region. At the cervical region called cervical flexure, that is the flexure between the spinal cord and hindbrain junction. And at the cephalic region called cephalic flexure, that will form the midbrain region. Thus, the neural axis doesn't remain straight. Like this picture, the forebrain at the midbrain region will form the cephalic flexure. So there is an angle between the forebrain and midbrain. This angle formed by the cephalic flexure. A second flexure at the rhombencephalon, between the rhombencephalon and the spinal cord, that is called the cervical flexure, will lead to angle between the midbrain, the brainstem, sorry, between the brainstem and the spinal cord. Thus, flexures is completed. The third step of development is ventricular system. The tubular structure of the developing central nervous system persists as development proceeds. The neural tube will have a cavity inside it, and this cavity leading to formation of what is called the ventricular system. In adult, it comprises interconnected reservoirs filled by CSF produced by the cells of ventricular lining. This CSF and the spaces inside the neural tube act as a cushion and prevent trauma and the spinal cord with their bony cases. The three brain vesicles on the caudal part that is from the spinal cord all have cavities inside them. The cavity inside the telencephalon will form the lateral ventricles. The cavity inside the diencephalon will form the third ventricle. The cavity inside the mesencephalon will lead to formation of cerebral aqueduct of Sylvius. And the cavity inside the hindbrain between pons and medulla anteriorly and cerebellum posteriorly will lead to formation of the fourth ventricle. The cavity inside the spinal cord will form the central canal. Abnormality of ventricular system development leading to some clinical diseases like hydrocephalus. Hydrocephalus is increasing amount of cerebrospinal fluid inside the brain ventricles. Most common in newborns suffering from spina bifida. It is always associated with spina bifida. The treatment of this condition by shunting process. Surgical treatment using shunt between the cerebrospinal fluid to body cavities like peritoneal cavity. And from the peritoneal cavity, it will be excreted by the kidneys. The cause of hydrocephalus, either by blockage of the ventricular system, like by tumor, infection, cytomegalovirus, toxoplasmosis, like this picture. This picture shows the severe hydrocephalus. See the size of the head. So the ventricular system is completed. Early organization of the neural tube. From inside, the neural tube divided into two tubercles, anterior and posterior. The anterior tubercle called roof plate and the posterior tubercle form floor plate. And the anterior plate will form the alar plate, the posterior plate will form the basal plate. From inside outside, the neuroepithelial layer and intermediate mantle layer. The neuroepithelial layer from inside and intermediate mantle layer that divided into alar plate and basal plate by a sulcus limitancy. The neuroblasts inside them are mesenchymal cells that have the ability to differentiate to different types of cells. And marginal layer by mesenchymal cells. These plates, also called the roof plate and floor plate. The alar plate will develop the sensory plate, and the basal plate will form the motor plate of the neural tube. This section is different at different levels of the neural tube. If we take a cross section at the mesencephalon, this picture, you can see, but on the metencephalon, the section is different. In the myelencephalon also, and in the spinal cord, it is completely different section. But the similar points of all these sections are the alar plate and basal plate. The alar plate will form the sensory plate posteriorly, and the basal plate will form the motor plate anteriorly. So this is the patterning and organization of the nervous system. Lastly, the neural crest, which are group of cells on the lateral border of the neurooctadermal part of the neural tube, become displaced. Then enter the mesoderm and undergo migration and changing from epithelial to mesenchymal transition. At first, they are epithelial cells, but undergo mesenchymal transition to become mesenchymal cells, which have the ability to differentiate into different types of cells. To understand this point, this is the neural fold. The edges of the neural fold form a group of cells called the neural crest. When the neural tube completely formed, these cells in the neural crest will enter the mesoderm, then migrate to different sides on the length of the body of the embryo and differentiate into different types of cells. They can give rise to cranial nerve ganglia, spinal root ganglia, sympathetic ganglia, parasympathetic ganglia, Schwann cells, glial cells, or meninges like arachnoid and pia mater. In the head and neck, they can lead to formation of connective tissue and bones of the face and the skull. Odontoblasts in the teeth, dermis in the face and neck, C cells of thyroid gland, and can lead to different tissues in the heart, forming conotruncal septum, in the skin, forming melanocyte, in the adrenal, forming adrenal medulla. Defects in neural crest migration will lead to extensive abnormalities of development. Complex migratory pattern, extremely vulnerable to environmental insult, especially alcohol, and can be genetic cause. The defects can affect single component, but can also affect multiple systems, resulting in multi-system syndromes. Conditions caused by defects of migration or morphogenesis affecting one structure like Hirschsprung disease. Hirschsprung disease is a ganglionic megacolon affecting the large intestine, forming a ganglionic colon, leading to huge volume of the colon, leading to abdominal distension, or affect multiple structures like DiGeorge syndrome, which include thyroid deficiency, immunodeficiency secondary to thymus defect, cardiac defects because the neural crest affecting the septum inside the heart, conotruncal septum, and abnormal facies due to abnormal development of the cartilages and bones of the face. And thank you very much.